Hello, everyone, and happy whatever day it is. What I'd like to do is sort of wrap up the Chapter 16 lecture. We started the first part on the structure of DNA. Now I'd like to get into the idea of DNA replication and some of the details related to that. So we talked about the semi-conservative model, where one strand serves as a template for the creation of a new strand or a daughter strand. Now, this will again differ between prokaryotes and eukaryotes. Prokaryotes have a single origin of replication, having transcription or replication, I should say, proceed in both directions until you basically complete the loop. And then eukaryotes will have multiple origins of replication on each of the chromosomes possessed by that particular species. So in the case of humans, we've got 46 chromosomes. Each chromosome will have multiple origins of replication. All right, that's sort of what that's saying. So now let's sort of look at the molecules related to this, the enzymes related to this. Helicase in green here is the enzyme that zips along the DNA molecule and breaks the hydrogen bonds between the nitrogenous bases. When that happens, it will sort of physically separate the strands, and then the single strand of binding proteins will clamp onto the DNA and keep the strands separated. When you separate the strands, that can create tension at the replication fork. That's the spot there where the strands are splitting. And so this enzyme, topoisomerase, will go and cut one strand of the DNA so that the other strand can spin on its chemical bond and unwind and relieve that tension. Then here we can see that we'll talk about this in a moment. We have another enzyme called primase that lays down this RNA primer. Oops, I'm jumping ahead here. You can take a moment to pause if need be to jot down information about these molecules involved. Again, helicase breaks the hydrogen bond, single strand of binding protein grabs the DNA and keeps the strand separated, and topoisomerase goes and cuts one of the strands to allow you to relieve that tension on the molecule. All right, so DNA polymerase. DNA polymerase, ACE tells it's an enzyme, makes a polymer of DNA. So this enzyme, like other enzymes, is really particular. It can add nucleotides all day long, but it can only add them to the 3' end of an existing molecule. So if we go back and look here, let's say here, if we look at the DNA molecule, remember the carbons are numbered. So here, if we look at the sugar, there's carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Now, the number 5 carbon, it's already got a partner, right? It's covalently bonded to this phosphate group. But if we go down to the other end, this 3' carbon, it's got the hydroxyl group, or this OH. And what this can do is be removed during a dehydration synthesis and form water. And so if you're going to be adding nucleotides, you're going to be adding to the 3' end of the molecule. So when we build DNA, star this, highlight this, underline it, whatever you got to do, just remember you always add to the 3' end. So because DNA polymerase, the molecule that makes DNA, is so particular, you actually have to lay down a short RNA primer first. So again, if we look at this image, DNA polymerase can add nucleotides and make DNA, but it has to add to the 5' end of something that's already there. So we have another enzyme called primase that can go and lay down a short segment of RNA primer. So it's sort of wild, but before we ever copy our DNA, the first thing we do is actually lay down a little strip of RNA. So primase will lay down like a 5 to 10 nucleotide long strip of RNA molecules, RNA nucleotides. And then that will leave a 3' end on the RNA segment. So then DNA polymerase can add to that, and we go and make the DNA. All right. Now that's all fine and good because there is another enzyme, another version of DNA polymerase that will go and remove those RNA nucleotides and replace them with DNA. So again, you can do that, but you need this other enzyme. Okay. So DNA polymerase is the enzyme. Now notice here that DNA, remember the strands are anti-parallel. One strand will run 5 to 3 and the other strand runs in 5 to 3 in the other direction. So because of that, when you build new DNA strands, they actually get built in different directions. So here, one strand of DNA, it's called the leading strand, is made in one piece and it's always growing toward the replication fork. So if you can imagine helicase zipping along and separating strands of DNA, as the replication fork continues to open, well, then DNA polymerase can just keep traveling along that leading strand and adding to the DNA nucleotide. But the other strand, it's called the lagging strand, it is growing in the opposite direction. It's actually growing away from the replication fork. So every time helicase unzips a new section of DNA, you have to lay down a new primer and DNA polymerase will grow away from the replication fork. Okay. So here is just a nice image sort of showing taking a new nucleotide and adding it in to the existing molecule. So again, we're always building or adding on the 3' end. Okay, DNA polymerase. It's a lot of words sort of reiterating things we've already talked about here. But again, the take home, always, always, always build 5 to 3. I just think of it as like going downhill, right? You're going from high to low or from 5' to 3'. But you always add nucleotides to the 3' end. And again, that leading strand is made in one piece traveling toward the replication fork. So here again, we see helicase separating the strands. Primase will add this little RNA primer, and then DNA polymerase will ultimately come in and add to the 3' end and grow in the direction of the replication fork. So the leading strand will be in one piece. Now, again, that lagging strand is built discontinuously. Every time a new section of DNA opens up in the replication fork, a new primer gets laid down and DNA polymerase grows the strand in the opposite directions. So those pieces are referred to as Okazaki fragments. So you'll create many fragments of DNA in the lagging strand, but then we have another enzyme called DNA ligase come in and form covalent bonds between the sugar and phosphate of the Okazaki fragments. So that basically stitches together the pieces into one continuous piece of DNA. So here we can look a little bit at what's going on with the lagging strand. The primer is laid down in red, and then here we see the primer is laid down in red. DNA polymerase will go in and add the bases, and then you create that Okazaki fragment. Then you open up a new strand, new primer, new DNA polymerase, a new fragment. And then eventually you'll have keep getting the bar along the bottom to show up. Down at the bottom there, what you'll do is you'll get ligase come in and it will stitch together those sugar phosphate backbones and you'll end up getting one continuous piece. So you can remove the RNA nucleotides, replace them with DNA, and then stitch all that stuff together. All right. In your book, in chapter 16, there's a nice summary looking at the enzymes involved in DNA replication and summarizing what they do. So if you would like some additional review, I think that's a nice idea to go back and just sort of review that, and then you can look at some of the images. So here is something like what you would draw in your chapter 16 activities. Here you see the leading strand, again, growing toward the replication fork. Here is your lagging strand, putting together or laying down primer and building these little Okazaki fragments. Here's another DNA polymerase, removing the RNA nucleotides, replacing them with DNA. And then, of course, you've got ligase stitching together those sugar phosphate backbones. All right. None of this is going to be testable material, but the basic idea is when we learn this stuff, we learn about the individual proteins and think of them like zipping along the DNA. In reality, the proteins tend to be clustered together into a big protein complex, and then you pull the DNA through. So rather than the enzyme sort of zipping along the DNA, you're actually extruding or pulling the DNA through and having all this stuff occurring. All right, so this is nice. Sometimes if you have damage or errors in the DNA, you can actually have proofreading enzymes that slide along the DNA and look for mismatches or problems. So one thing that can happen is you can get nitrogenous bases dimerize. So if we look up at the top here, The nitrogenous bases can actually, rather than having their nucleotide partner and then they hydrogen bond with a neighboring nucleotide, what they can actually do is form covalent bonds with one another, and then that sort of creates like a kink in the DNA. So then when you go to want to transcribe the DNA, it could mess that up. So we have these enzymes like nucleases do what's called excision repair. They'll actually cut out that section of DNA that's got the mutations of the errors, and then DNA polymerase can come in and add in the correct bases, and then ligase can stitch it back together. So you actually have mistakes occur all the time in your DNA replication, but you have enzymes that go and fix those errors. So we can repair our mistakes. So that's pretty remarkable. All right. So again, we've got just recognize the fact that we've got enzymes that can go and proofread the DNA, find mistakes, cut them out, and then replace them. So lots of words on that slide. I would just summarize it like that. Right? They're enzymes to identify mistakes in the DNA, so they can cut out the mistakes. You can replace the incorrect nucleotides, and then you can stitch it all together. If you've got that, you got the concept. You're good. Okay, now here's getting into why we must all die. All right. So we know that you always build 5 to 3 prime, right? So we have linear chromosomes. They're long threads or lines. So when you get to the very end of the lagging strand, you'll have your, primer laid down. But then when you get to the very end of the molecule is 5 prime, right? Well, what can DNA polymerase attach to? Nothing. So because of that, the primer at the end of a replicated strand can't have its bases replaced by DNA polymerase. So in essence, every time you copy the DNA, the ends just get a little bit shorter and shorter and shorter. The ends of chromosomes are called telomeres. And so what happens with each successive round of cell division is that the telomeres shorten more and more and more. Now, generally, it's not a big deal because remember, in your chromosomes, only like 1% of all those A's, T's, and C's and G's code for genes. So the ends of your chromosomes have A's, T's, C's, and G's, but they're not genes, right? They're not instructions for making proteins. So you can have your telomeres shorten and you're totally fine. Your genes are still intact. But if they shorten too much, well, then you can start getting in the genes, and then the cells will perform apoptosis. So here's a nice image of this. Here you see and this is in the lagging strand here. You have the RNA primer laid down, and then as long as there's a 3' end of the molecule, well, DNA polymerase can go and replace that. But if we get to the very end, DNA polymerase can't attach anything to this 5' carbon, and so that portion does not get replicated. So with each successive round of cell division, again, telomeres shorten. Now, what that means is normally cells are only good for around 50 rounds of cell division. So after about 50 rounds of cell division, they're not going to divide anymore, and then that is that. Okay, so our bodies only have so many rounds of cell division in them. So telomere shortening is something that's also sort of looked at as related to biological aging or genetic aging or chromosomal aging, however you want to think of it. Okay. So again, the ends of our chromosomes, telo means end. So the ends of our chromosomes shorten with successive rounds of cell division. And so, of course, that sort of ties into cellular aging. Okay. So, it's sort of an inevitability for all of us. Maybe. Okay. One thing that's been found in some small scale studies is that if people do a really holistic approach to self-care, so if they exercise, I forget what the time was, maybe 60 minutes a day. So if they exercise daily, if they have small group sort of discussions where they're sharing problems and discussing things that are going on in their lives, they practice things like meditation, and they eat well and get enough sleep, some people in these studies actually showed lengthening of their telomeres. So really good and consistent self-care can actually be associated with reversing some of this telomere shortening that occurs. Now, it doesn't mean you can stay young indefinitely forever, but you can sort of slow this progression. And of course, you can imagine there are already products out on the market that advertise the fact that they can lengthen telomeres. I've seen commercials before where companies are advertising stuff. I wouldn't go buying anything like that, but it's actually been discussed. And one thing that's sort of interesting is, we know that when you copy genes with making gametes, wouldn't you think that the chromosomes would get shorter and eventually, your gametes wouldn't have enough DNA? Well, that's, a reasonable thought. So, of course, there's an enzyme for that, telomerase. Telomerase is an enzyme that basically adds A's, T's, C's, and G's to the ends of these molecules so that when you're making gametes, after successive generations, the chromosomes don't get too short. So, of course, some people are like, well, hey, couldn't we do something with telomerase activation to potentially see if we can sort of reverse some of this cellular aging that occurs? And that's a great idea. Of course, the thing you have to think about is if you can have chromosomes that can be maintained over many rounds of cell division, well, what about the accumulation of damage and then the ideas related to cancer? And actually, there are lots of examples of how telomerase is switched on in certain types of cancers. So cancer cells can divide and divide, divide and divide and divide, and then keep going because they have telomerase to prevent their chromosomes from shortening too much. So there's actually a chemo drug that targets telomerase so that the cancer cells can divide and divide and divide and then sort of peter out and die. All right. So again, College Board loves comparing and contrasting. So we have bacterial chromosomes. Again, it's one chromosome and it's circular. And then eukaryotes like us, we have linear chromosomes. They're like threads, and we'll have many of them. So that's good to compare and contrast that. Chromatin is DNA wrapped around these histone proteins. So that's sort of a nice image that we see here. The DNA has been untwisted, and then it twists up. It wraps around these histone proteins, and then it packs in really tightly. Okay. So here just sort of shows the successive folding of DNA into chromosomes. So if you took all 46 chromosomes in a person and unwound them fully, it'd be literally 6 feet long. So we can pack 6 feet of material into something that you can't see without a microscope. So that's pretty cool. All right. And hey, you made it to the last slide. We're going to talk about the different forms of DNA. If DNA is all coiled up really tightly, it's referred to as heterochromatin. So that's what we see on the ends of this image here. And then if the DNA is more loosely packed, it's referred to as euchromatin. U meaning good or true. So with euchromatin, I think, hey, that's the good stuff. That's the DNA that's more loosely packed. So if you look at it, well, hey, you can get at that code, right? So when DNA is relatively relaxed, you can separate the strands, get at the code, use it to make proteins. If the DNA is packed up or condensed into heterochromatin, the other stuff, then you can't get the enzymes in there and you shut things down. So what's really wild is for some genes with things that happen embryologically, and we think about like imprinting, like genetic imprinting, you can permanently shut down some sections of DNA, some genes in an embryo, and then they stay shut permanently. Then with like X chromosome inactivation in females, one of the X chromosomes randomly gets shut down. And the thing that's even wilder is in many genes and cells throughout our body, the stuff we do throughout the day actually causes, it's called a methylation or acetylation, DNA to coil up and uncoil. So the things you do throughout your day, exercising, smoking, getting enough sleep, what you're eating, the stress you experience can actually change the shape and ultimate expression of your genes. So, in the Mendelian stuff, we learn that it's important to inherit certain genes, but that's really only part of the story. It's what you do with those genes, because what you do during your life can actually affect the packing of that DNA, and you can alter your gene expression through the things you do. Okay. So genetics sort of sets the range for what is possible in your body. And then the things we do allow us to sort of drive the expression of our genes in one direction or another. So your genes are definitely part of the story, but it's how we sort of carry out our day-to-day lives that allow us to sort of express ourselves, at least in terms of protein production. All right, so that's what we've got for chapter 16. If you have any questions, let me know. Otherwise, I hope you have a great day and take care.